So, hello everyone. We are once more in the What We Are Reading YouTube channel of the South American Mycorrhizal Research Network. I'm Cesar Marin, and today we have with us Rebecca Bunn from Western Washington University. She's a professor there, and Ana Corrêa from the University of Lisbon, together with a really nice team of all female researchers. They published recently, very recently, a very nice Tansley review in New Phytologist about a topic that I think that we all mycorrhizal researchers wonder about, which is how actually carbon is transferred to the fungus. We will link the Tansley review in the description of the video. And with that, I give the word to Pedro Antunes, who will make the first question.
Hey, hi everyone, and yeah, congratulations on your paper. It was very, very interesting. And it's one of those papers that changes or really helps us think about something we sometimes take at face value on how this symbiosis is maintained and how it evolved. And so yeah, it was great to read it and think about it more deeply. And my first question was actually, how did it come about, right? So what's like the behind the scenes? How did the team come together and why? Could you tell us a little bit more about that?
Yeah, I think I can take the lead on that. And I just also want to say thanks for the opportunity to talk about this work, which I think we can honestly say we feel very passionate about and really has changed the way I think about the symbiosis as well. So this actually started out with neither Anna nor I. What I've been told is that Cindy Prescott reached out to Justine Karst and said, let's form a working group to think about how resources move between mycorrhizas. And that started to grow organically by invitation. And I know Ilva Leckberg joined, and I think Anna Sala, the plant physiologist. And they wrote a query to new phytologists and suggested doing a Tansley review. And received an invitation for that Tansley review back in November of 2022. So that's when they reached out to a few more people, and I joined the group, and Anna joined the group, and other people joined the group as well. It actually kind of grew and shrunk a little bit. So over time, we just started meeting and people sharing their own ideas and own work around what governs resource transfer between plants and fungi. And we would meet over Zoom. We were meeting every other week, really, for an hour, or sometimes it'd be 2 hours, and grow on beyond that. There were some really frustrating meetings. And I think the first deadline for the review came and went without us even having an idea what we were talking about. So we just really talked in circles, realizing that we had a lot of terminology to work out. So things we would use the same word, but mean different things because we were coming from different backgrounds. And also really wrestling with the realization that the outcomes of these different frameworks overlapped. So we would sort of have our own justification and then present our evidence, and the other person would come in and say, but that could be explained by my idea as well. So that really had us turn back to like, where did these ideas come from and what are we talking about? And another piece we really struggled with was, were we talking about evolutionary timescales, or were we really talking about what's happening right now in a physiological experiment? Because those are different kinds of evidence. So we finally had kind of a reason to dive. We dove into the literature to see like, where are these ideas really come from, and where are the overlaps, and how can we kind of figure out what we're even talking about, and let's define the terms. So that was sort of the first piece of our work. And then we looked at the experimental data and tried to understand which of these frameworks was supported or what aspects were supported. And again, we started out with this idea we would just be directly contrasting biological market models with surplus carbon, but it really did not turn out that way. So we felt like at the end, the evidence, the data, experimental data in the literature couldn't really tell us which of these frameworks was appropriate or was most useful. And that we really had to get at this idea of whether resources moving across the membranes were directly coupled. And that's what took us to look into the molecular mechanisms, which is an area where there's a lot of questions still. It's so again, we couldn't find the answers there, but we felt like we were getting closer. So that was kind of the second set of work, and then moving into what, like the future of what we thought would happen going forward. So yes, we continued to meet, but maybe like a year ago, we finally started to make some progress on what we're really writing about. And a second deadline went by without any paper. And as the third deadline was coming up, we finally were like down to the wire, but we were still like going back and forth right up to when I submitted it. And I think it was June 1st. And so, yeah, it just really, even at the end, we had some disagreements, but we feel like the paper's written in a way that allows for those all to be held, because I think we still have quite a bit of work to do to answer this question. So yeah, that was kind of the trajectory. And there were some people like Roger Coity was part of our group, and then he stepped out and he's acknowledged, but not in the authorship by his own choice. So yeah, that is kind of the whole arc of what happened.
Thank you so much. And the article has a nice glossary, which is very useful. I invite our listeners to go and read the glossary. But can you, and any of you, please explain us what in a mycorrhizal context, what a biological market is, and between the concept of a biological market is the concept of price. So what is the price paid by the fungus and the plant in this context?
You take this
Anna,
one
do you
also?
want to take this? Oh, I can take this as well, and then you can do the next one. So a biological market is where we have partners of different trader classes. So in the mycorrhizal context, this would be our plants and our fungi. So plants are better at fixing carbon and making carbohydrates, and fungi are better at acquiring mineral nutrients. So we have these two different trader classes, and those partners then trade with each other for the other resource. So the price here in the mycorrhizal symbiosis has to be, because plants and fungi don't have any common currency, it's that exchange of carbons per unit of nutrient. So you could think of it as how many units of carbon are spent per unit of resource returned. And so that exchange rate is then the price under the hat of a biological market model.
Yeah, I would only like to add that they have been applied in two different ways. So there are comparative advantage models where this exchange rate is used to explain degrees of benefit. So when the conditions are more or less beneficial, and so they explain the outcome of the symbiosis. And then there's help me with this one, partner choice. And those, they use the exchange rate, they propose that the exchange rate is regulatory. It regulates the exchange.
So actually, this is one good, I just want to add one thing here. So one of the terms we had a hard time with was partner choice versus sanctions and rewards. So partner choice has actually been used both to mean choosing a fungal partner, like a plant choosing a fungal partner to form a symbiosis with, and it has also been used in the literature to mean directing resources to one fungal partner over another when plants have options. And that second definition really overlaps with the idea of sanctions and rewards, which is part of these market models. So the sanctions and rewards idea is that if a plant is not receiving anything from the fungus, it would cut off the supply of carbon. If it's receiving a lot, then it returns a lot. So we're actually suggesting that partner choice should be reserved for that initial colonization and not be confused with sanctions and rewards. So Anna, I would say that second
It's
model we're
sanctions
talking
and
about more
rewards.
is sanctions and
It's
rewards.
sanctions and rewards is yeah. My God.
Right. So I guess that if we're thinking about sanctions and rewards is more in terms of evolutionary biology and what controls and maintains the symbiosis. But if we're thinking about the other component of or the way of thinking about this, we're thinking about sinks here. And what constitutes a sink in this case? How would you define a sink?
So we're talking about sink strength. So in the fungal case, it would be all the fungal carbon needs. So this would mean respiration, all carbon used in all metabolism, carbon used in growth. So it's really how much carbon the fungus is needing for all of its needs at a given point. Well, the same with the plant, but here the relevant one would be fungal carbon sink strength, right? Because we're talking about movement of carbon from the plant to the fungus.
Mm-hmm. Yeah, that is that was also my understanding. And so then I guess this leads right away to the next question. So I'll move with that. So then what's the empirical actually no, sorry, what do you mean by co-regulation here of carbon and nutrient transfer? How is that exchange controlled? Who controls that exchange? What partner?
Okay, so that depends on the hypothesis, right? So under biological market theory, and this is not really explicit in the proposal, but after a lot of discussion, we came to this conclusion, which is that the theory, both hypotheses, surplus carbon and BMT, the big difference is here. It's what regulates exchanges. Everything else, they can accommodate the same concepts. Everywhere else, there can be overlap, but here they clearly they are clearly distinguished. So under BMT, under biological market theory, and sanctions and rewards actually, because comparative advantage doesn't go there, there seems to be an assumption that the regulation is local. So how much carbon is going to the fungus directly depends on how much nutrients are being received from the fungus at the local level. And so whatever regulation mechanisms are in place, they are local and they depend on the exchange rate. So carbon transfer will depend on nutrient uptake. Under surplus carbon, this does not happen. So exchange rates do exist because exchanges are taking place. So of course, there's always an exchange rate and they vary, but they are not regulatory. So what will determine that the plant is transferring carbon to the fungus is really the nutrient status of the plant. So the more nutrient limited the plant is, the more is photosynthesizing in surplus of what it can use for its own growth and its own metabolism. So the more carbon is available to then go be transferred to the fungus. On the fungal side, and here we're extrapolating, we have nothing on the fungal side, right? We don't really look at the fungus, but the same thing can be happening. So the more carbon limited the fungus is, the more nutrients, because the fungus is taking nutrients. If it's more carbon limited, it's not going to be able to use all the nutrients, and those nutrients are the ones that are going to the plant. So it's more of a systemic regulation, and it does not depend on the resources that are being received back.
Okay.
So
So
can I add one thing?
Yeah, yeah.
Just to,
yeah,
yeah. So I think Anna, you said really nicely, like no matter which of these frameworks is operating, we see certain amount of carbon going one way and a certain amount of nutrients going the other. So that's where there's overlap. But under that sanctions and rewards, we were really looking for some molecular mechanism where carbon was released in one direction when nutrients were transferred in this direction, like something that really coupled that transfer across the membranes, as opposed to just the, we know these flows are happening.
yeah. So this is really like the distinction is that, right? That's and then under BMT, the regulation has to be local. So we were looking directly at what, yeah, you already said this, sorry.
So by local
Oh, yes,
you mean like in
symbiotic interface. It's really symbiotic interface, so it's membrane, yeah.
okay, good.
Mm.
And then that brings me to like I think a very important question which you discuss very nicely across the Tansley review, which is the empirical evidence for both models, for the surplus C hypothesis, surplus carbon hypothesis, and for the biological market models. And for example, something that we all wonder about, which is how really selective are plants by choosing or not which mycorrhizal fungi they associate with.
I can briefly talk about the empirical evidence that we summarized in the Tansley review around the trading of resources for the vote counting, which I think Cesar will get at that question. So Ilva did a nice vote counting exercise where she looked at, in Anna, they extracted the results from studies that had plants growing with multiple fungal partners and tracked carbon and nutrients moving with the question of whether plants were preferentially rewarding better symbionts. And by better symbionts, we mean fungal symbionts that are returning more nutrients. And found that there was, just from vote counting, it wasn't, the majority of studies did not show that plants reward better fungal symbionts. But also there was no way to determine whether better fungal symbionts were really just ones that were more metabolically active. So this again was a big overlap in these frameworks. Both the biological market models and the surplus carbon models would predict that more carbon flows to more metabolically active fungi and more nutrients flow to more photosynthetically active plants. So these experiments where we're tracking flows aren't actually giving us an idea about which of those models is more useful. Anna?
So it's like in more specific terms, if we think that we're giving two fungi, one has more access to more phosphorus than the other, and more carbon is going to the fungus which has access to more phosphorus, we can say that that fungus is giving more phosphorus to the plant, which it is, but it's also more metabolically active, right? It's taking up more phosphorus, it's growing more, everything. So its carbon sink is bigger. And as long as we test these things like this, as long as we use this kind of experimental setup, we're never going to be able to differentiate sink strength from nutrient transfer, like nutrient, how much the fungus is giving nutrients to the plant. I think can you repeat the question, actually? This was a part of it, what was the other?
Yeah, well, which is the empirical evidence for both frameworks, for surplus hypothesis and I think that you partly answered.
Yeah.
Well,
we felt like at the end we didn't find any strong, convincing evidence for biological market models that we couldn't interpret in another way.
for surplus carbon, it's a bit different because surplus carbon came from empirical evidence. So with BMT, what you have is a conceptual framework and theories coming from economical theories being applied. So, and then looking for evidence to support or not that, or like finding different ways to use them. With surplus carbon, the empirical evidence came first. So it arose from us seeing that the carbon transfer to the fungus is not costly. It's not responsible for growth depression in plants. So to begin with, one of the big, then there's different aspects of the hypothesis, but one of the big ones came from empirical evidence, which is carbon is not costly, the carbon which is being transferred. And then, okay, so let's see if I don't forget anything. So then there were different aspects that derived from there. One was that what is regulating the carbon transfer, which I already mentioned, and the idea that is not nutrients coming from the fungus, it is the nutrient status of the plant. And that is supported by the fact that plants will form more mycorrhiza the more nutrient limited they are, and regardless of how much nutrients they're getting. So this does not depend on nutrient benefits. It only depends on nutrient deficit from the plant, right? So, and that was also supported. And then the other thing that derived from there was this idea that if we don't see that the nutrients are regulating these exchanges, then maybe, and all the mechanisms we're seeing, although indirectly, seem to be the same as are already in place for non-mycorrhizal plants, then maybe everything we know about the non-mycorrhizal plant also applies to mycorrhizal plant. The mechanisms are the same. They're not mycorrhizal specific. And we don't know, there's a lot we don't know here, okay? So we have a lot of work to do. But so far, we could not find anything. And so carbon transfer, all the transporters that are involved are not mycorrhizal specific. So they are also active in non-mycorrhizal plants. That's one thing. And they may be more active in mycorrhiza, but they are not just in mycorrhiza, which is the case for phosphorus, right? For example, phosphorus in AM, there's mycorrhiza-specific phosphorus transporters. This is not the case for sugar. And the fact that none of the sugar transporters that we know of are regulated by nutrients. So that link also doesn't exist. So yeah, so that would be the surplus carbon part of the empirical evidence.
Okay, so if partner reward is not doesn't fit, it's not a thing, then what other mechanisms could be involved then to determine partner choice, host specificity? Plants associate with different mycorrhizal fungi, different types, and so what mechanisms could be involved here in that type of partner choice?
Can I
Yeah,
start
yeah,
and then
go.
yeah. So I just also want to emphasize the fact that we haven't proven biological market models. Doesn't mean there isn't still room for that model to be useful. And so at the end of our paper, we talk about some areas. So one of the things that came up there was spatial scales. So let's not throw them, we don't need to throw them out, I think, but thinking about what mechanisms might be in place, a lot of what we've observed, everything we've observed so far could be explained by by metabolism of these organisms, right? So again, what I said before, all of these frameworks accommodate that the flows, if they're going from source to sink, is one explanation. So when a plant is very active and photosynthesizing, it becomes nutrient limited. Carbon flows to the fungus, which becomes metabolically active, and it's the biggest sink, so it's receiving the most carbon. The other explanation is that fungus is returning a lot of nutrients because it's so metabolically active, and so plants are allocating. The problem with allocation is just it's more complicated. It requires the plant to recognize the nutrients that are coming and direct flows towards it, as opposed to the simple explanation of physics, right? Where we're flowing from high concentration to low concentration. So I think, like Anna said, we still have a lot to learn, but the idea that this is governed more by physics than by intention and signaling is a simpler explanation.
It would be really about sink strength and about how nutrient or carbon limit the organisms are and how much surplus resources they have that they can just give away. So it could be really a simpler, but I think your question touches another thing, which is there is fungal selection. I mean, we know that it exists. Plants form mycorrhizae with some fungal species than with others. They prefer some ecotypes. If you have a nutrient gradient in in an ecosystem, you will have different fungi forming mycorrhizae depending on nutrient gradient. So that does take place. We don't know how, and we don't know what factors are involved. I think we know very little about what determines this. But I think here we can go to this distinction between sanctions and rewards and partner choice. So partner choice can be something that takes place at the molecular level, I don't know, immune system, something that somehow, we don't know how, the plant recognizes that this would be the appropriate fungus for now. We don't really know how this takes place. But then there's the maintenance of that symbiosis, of that interaction, and the maintenance can be just governed by sink strength and nutrient limitations or nutrient needs. And there's another aspect to this, which is there's a difference between carbon transfer and nutrient transfer, in the sense that carbon transfer, like sugar transport, seems to be very unspecific. And the main transporters involved in carbon transfer seem to be sweets, which are channels. They're not everything. All of this is not just pure physics, because there's always regulation. But sweets are really their channels, so they function with concentration gradients. So this is sugars, but for phosphorus, for nitrogen, things are more specific. So it's not that we can completely rule out that nutrients are a part of this regulation. It's the link between carbon and nitrogen, carbon and nutrients, which is missing. So maybe somehow the plant recognizes that this fungus will be a better partner for phosphorus, but then how much carbon goes to the fungus is now independent of how much phosphorus it receives back. It's just happening.
So
I think I get it. Sorry, yeah, Cesar. I just had a quick follow-up question, if that's okay. So I think I understand exactly what you're saying. And so this sink strength, and there's no regulation of carbon, but there could be regulation by these other types of mechanisms that you mentioned. I think in your paper you talk about some mechanisms that are similar to pathogens, and those would you say that those kick in more strongly when, for example, nutrient availability in soil is higher. So if phosphorus is really high,
it's a huge question. That was a recurring question, actually. We have no answer for that. Like, because we do know that plants form more mycorrhizal, the more nutrient limited they are, and the other way around, right? If they can't take nutrients directly through the roots, they will not. They will stop the carbon flow to the fungus. What determines this? Where is the switch where a plant goes, oh, okay, carbon goes to root growth, or carbon just goes to direct nutrient uptake? This is a huge missing link. We have no question. We have no answer for that one.
thank you.
When Anna, when you were explaining about how both frameworks came about, it was very interesting to me because you mentioned that the surplus carbon hypothesis comes from empirical evidence to put the framework out, while biological market models have the opposite direction. And this is basically a difference between deductive and inductive science. So one framework is very deductive and the other one is very inductive. And recently, I think this year, there is a paper in Evolution, which is, the title is like Popperian ecology is a delusion, meaning that the deductive ecology is not so realistic. That the paper argues that in ecology we think that we are doing like Popperian ecology, but we are really doing induction. So seeing the empirical evidence and then propose frameworks or hypotheses or ideas. So this is very interesting to me. And the second thing that I think that both frameworks, but correct me if I am wrong, but both frameworks might have in common is that increase fertilization, increase nitrogen and phosphorus leads to less mutualism, or leads to less colonization or less arbuscules formation, right? But it seems like both explain that through different mechanisms.
Okay, with surplus carbon, it would be the same thing, right? Like it would be, because it's always the same proposal. If the plant has more nutrients, it can use more of the carbon. It's more of the sugars it is photosynthesizing, so less carbon is available to go to the fungus. That's it. I have to think about what would be BMT's take on this.
So the price of nutrients would go down, right? So you don't have to send as much carbon to get more nutrients, and so you can use more of that carbon for growth, and you're getting more nutrients. So it's less of a cost to acquire nutrients when there are a lot of nutrients, because your market is flooded with nutrients.
Oh yeah, okay, so it would be if the plant can, okay, now taking nutrients directly through the root would be less carbon expensive, so the plant will do that instead of taking it from the fungus. Because it doesn't really specify, right? Like the BMT doesn't specify an explanation for why the less mycorrhizae is formed with more nutrients.
I think it'd be because you don't have to spend as much carbon, so you just don't have as much fungus, right? You're getting more nutrients for every unit of carbon that you're sending.
Yeah.
Yeah, and with that,
oh yeah.
Yeah, sorry. And with that, I will ask one more question, and I will lead Pedro the final, like, more general question. My final question is related to kind of the molecular and metabolic section of the review, which again, correct me if I am interpreting this in a wrong way, but it seems to me like yeah, but definitely mycorrhizal fungi. Um, the question is, should mycorrhizal fungi be considered extensions of the roots? And it seems like all these metabolic writing that you did and the molecular mechanisms leads to that.
I think I can just say that we disagree. This is a point where the authors disagreed, and that some of us felt strongly that when you have two different organisms, there are boundaries at those membranes, and so accounting for resources moving across each boundary and what regulates that is the way forward. But then Anna can present the other side.
Yeah, it's actually nice because we have two sides represented here. Yeah, I would say yes. On the fungal side, very clearly. I mean, especially for our vascular mycorrhizae, they can't synthesize lipids. They need the plant. So there's a metabolic continuum very clear. Then there's functional continuum for sure. And how intimate like how much of a continuum they are. Still, we need to know more about what happens in terms of mechanisms. So for the fungi, it's more clear because, of course, lipids with AM, but even with ectomycorrhizae, unless I'm mistaken, something has been published that I don't know about. Ectomycorrhizal fungi do not form fruiting bodies if they are not in symbiosis, right? I hope I'm not saying anything that is no longer true. So there is a dependence there. And of course, our vascular mycorrhizae cannot grow. They have no independent life. For the plant, this is less clear because the plant can live without the fungi, mostly, if they have enough nutrients and so on. But even if we don't, I think purely as an exercise, we should consider this possibility. Like, what changes if we think of these organisms as functioning in continuum instead of these organisms being separate and always in constant negotiation, and with a clear border? So if for nothing else, just as a way of looking, because this will change how we understand and how we study these interactions. So if for nothing else, we should at least consider this possibility.
Yeah, that leads me to one more question, actually. So yeah, I think that your proposed model, carbon surplus model, is very well supported in the paper from anywhere you try to look at it, as you explained very well. And so it really releases us, by us meaning everyone in the field, from thinking more strictly about reciprocal rewards to considering a lot of other things. And what are these a lot of other things? How do you think that this paper that you've just published and these new ideas will now contribute to advancing the field? What kinds of things does this release us to explore now?
I could take a stab at it, and then Anna, you can fill in. Yeah, so for us, just because we struggled so much at the beginning to even have a conversation about resource movement, we literally were using the same words and meaning different things. We really hope by laying out specific definitions for the terms, having clarity around how these concepts arose, where they came from, and appreciation for how much the predictions of these different frameworks overlap. We hope that will allow people to kind of step back, researchers to step back and really ask what they are asking and what they're testing in their experiments, so that future research can be more productive. We also hope this really highlights how important it is to work across scales. So I do think the questions we've been asking, I'm more of a molecular or a mycorrhizal ecologist, really can't be answered by the kinds of studies that I usually do, which are at the organismal level. And we really need to collaborate with people across scales, for instance, molecular ecologists or biochemists, to understand some of these, to answer some of these questions. And the last thing I really feel like this made me do, I came in really under the biological market model framework. That was my thinking, that plants, this cost benefit, and that carbon was costly to plants. And it was really hard to look at that and change my mind. So I hope this paper also helps researchers step back and ask what assumptions or potential biases they're bringing to their work, which will free them up to more directly ask questions that they can answer. So Anna?
Yeah, I would only add that to that. Yeah, well, just reinforcing what you just said, like we are using a lot of models and conceptual frameworks and thinking without really understanding. We're just taking them for granted and we're just using them, and that introduces biases. But also, I think it was really important to identify where we are not researching anymore. What are we not looking at anymore? Like specifically for physiology work, which was until 2000 maybe, or the early 2000s, it was very active and then it stopped. And we don't know a lot. We don't look at, maybe we still look a little bit at transporters, but even that has slowed down, and we don't really know what's happening in terms of physiology. And that's important. We need to go back there. Yeah, and because it was so much hard work for us to understand what do we need to test for if we are going to find evidence for one or the other framework. That's really important also.
Yeah, thank you for that. Yeah, leads me to so many other questions I had about where did this biological markets came from exactly, did it come from, for example, more host-specific types of symbiosis like rhizobium, and as opposed to some less host-specific, where maybe the zinc strength might be just a simpler way to explain. So it leads to so many questions I still have, but we would be here maybe all day. Cesar, do you have any other questions?
No, many thanks to both of you and to Pedro for your time, and yeah, as always, the invitation is to actually go and read the whole article, and yeah, we will keep posting these interviews. And yeah, what I mentioned before, just seeing how you contrast both frameworks and other frameworks, and seeing the assumptions is like philosophers of biology would have a field day checking this, because it's really nice. It's like a really real life um experience on how paradigms shift, and how actually science is done. So I am really much enjoying this without taking one side or the other. Just to seeing the field develop is really nice. Thank you so much.
Thank you. Thanks for the
Thank you.
opportunity to share. Yeah.
Yeah,
Bye.
thank you.